welcome everybody. I see we have 56 participants on today's webinar, and that's fantastic. This is the first of a series of webinars around engineering biology that we're going to be conducting over the next few weeks. And again, I want to thank all the speakers who've agreed to present, as well as the participants who've signed up. The history of this event is due to a conversation that I had with the Dean of Ag Bio at the University of Saskatchewan. And she was asking if we were going to do a seminar on what is engineering biology and increase awareness of the platform. And we've decided to actually not have me give a seminar, but to actually have experts provide seminar information on engineering biology. And our first speaker today is Dr. Patrick Westfall. And we have, Patrick and I, full disclosure, have previously worked before at Dow AgroSciences, now Corteva. And Patrick was a group leader and instrumental in helping our organization apply the automated build to answer key questions related to plant genotyping, or gene editing, I mean. And again, I think it's a great example of how the technology enables research. And Patrick is going to give us an introduction into engineering biology and its how it works, what it is, and its potential applications. And he's got some wonderful experience. And again, he did his bachelor's at Purdue University, and I'm going to stop there because he, being a Hoosier and
Boilermaker.
That's right. I meant from the state of Indiana, and I just stepped in the biggest mistake. And Kirk Brewster, if he's on the call, is going to disown me because, again, a great school. The opportunity for Patrick to kind of introduce himself, his history, because again, he's got a lot of experience in this space. And I think it's going to be really valuable for all the participants to kind of get Patrick's experiences and perspectives on the evolution of the technology and its application across various business verticals, including ag and food. So Patrick, I'm going to hand it over to you because they don't want to hear me talk.
Great. Thank you, Steve. I appreciate it. Okay, so everybody hopefully can see my slide deck. Give me a thumbs up if you can see it, Steve. All right, we're good to go. If you look at the title slide, when I interviewed back at Dow, I gave a talk about, kind of interchangeable parts. And I used an example of Legos as one of my favorite toys as a kid. And that was why there was a Lego Millennium Falcon up there. I think that actually came from my job talk at Dow. I'm going to go with a different toy from my experience here in a second. That's Newton's Cradle. Before I get into that, though, I kind of thought I'd introduce myself and kind of how I ended up here, giving this talk and what I've done. So we have the Grateful Dead bears there. So I did have some experience at Berkeley at one point in my life, and from trucking, what a long, strange trip it's been. So I started out as an undergrad at Purdue, studying genetics, studying fungal biology, actually. And from there, I went to the University of Georgia, where I studied plant pathology and actually my PhD in botany. So plant biology was kind of where I started out, and then fungal and microbial biology is where I went. From there, for my postdoc, I started out at UCSF, working with one of the preeminent yeast geneticists, Ira Herskowitz, before he passed away. And then I finished my postdoc over at Berkeley, thus the Grateful Dead bears there. From there, my first job in industry was working for a synthetic biology, engineering biology company called Amyris Biotechnology. And at the time, they had 29 E. coli geneticists, and I was the lone yeast geneticist. And I was able to join Amyris and lead a couple of pretty amazing programs as a scientist and grow the first part of my career there working on engineering yeast for making of terpenes. So basically taking plant pathways out of plants and putting them into microbes like yeast, and then making plant products, things like malaria drugs or biofuels from the plant genes into fermentation and Saccharomyces. At that point, I joined in 2012. I joined Dow AgroSciences, where I worked with Steve. And this was my first foray into genome editing. So at the time, the hottest technology was zinc finger nucleases. And it doesn't seem like it was that long ago when that was the cutting edge. Of course, with CRISPR now, that's not quite as important as it used to be, but it was my first foray into how do we put, genes into recalcitrant organisms. And it really has been that the advent of genome editing technology, our ability to manipulate recalcitrant organisms, has really been the step forward I've seen in my career. That's really been the game changer of how we proceed forward with biology and engineering. From there, I went to back to the Bay Area. I joined a company called Zymergen, which was another synthetic biology company started by a couple of people who left Amyris. And whereas Amyris was really a yeast and E. coli shop, Zymergen's approach was to work with all organisms, no matter how difficult they were. And so I was there for about 4.5 years before I took my current job at Inscripta. And I've been here since February of 2020. Really working back in the genome editing space. And all the previous jobs, I'd always been working on products. And Inscripta has been a real treat because I've got a chance to work on tool building and think about how we can take synthetic biology engineering, and really make an attempt to democratize it so that it's fast, it's easy, and it's accessible to anybody who wants to do it. So like I said, my job talk at Dow was using Legos. I'm going to use a different toy here, Newton's Cradle. I still remember in my house growing up in our basement, my dad had a little wet bar set aside, and he had a Newton's Cradle sitting on the wet bar. And I would sit there and, we'd drive my parents nuts because we clicked the ball back and forth, back and forth, back and forth, and showing that conservation of motion. And I started thinking about this talk and how does this concept fit into how we approach biology. And so this is the model I came up with, is that, throughout my career working in industry, working in academia, we really kind of work at two ends of the spectrum, and engineering biology covers both of these spectrums. And at the one end, you have empirical engineering. This is things where you don't know the answer, you're just looking for solutions. This is how it existed before the advent of molecular biology, before the advent of DNA manipulation. Is you'd find something and you'd mutate it and you'd look for improvement. You'd screen, but not really know what the mechanism behind it is. And then about 2000, as we started to understand genomes, I started to understand genetic engineering, we started driving ourselves to a more hypothesis-driven engineering approach where we'd put promoters or genes or pathways in. We were much more intentional. We knew what to expect from that. And we still work at these two extremes, but the problem is you can actually back yourself in a corner if you only look at them exclusively. And so for this model, I wanted to use the motion, the steel balls that are part of Newton's Cradle, and think about how we work at either ends of these approaches, whether it be radical empirical engineering or hypothesis-driven engineering. And that center ball of Newton's Cradle is where we all start out with, is this core knowledge. This idea that we understand the organism we're working with. Is it yeast? Is it corn? Is it canola? Is it E. coli? We understand the physiology of that organism, what it takes to grow that organism, what it takes to propagate that organism. And we understand metabolic pathways. We understand the conservation, understand things like kinase cascades and enzymology and how that works. But there's that core knowledge of the organism and the metabolic pathways we start out with. And that's pretty solid. And you'll notice in Newton's Cradle, that's not the ball that moves. That is the core to everything that we do. But then you move out, and you move out to the two steel balls on either side. And now you start to see, you can take approaches that are more hypothesis driven or more irrational. And on the irrational side, now we start to go to assumptions, things that we think we know, but we maybe don't actually prove. So one of the most common ones I've seen is toxicity. So you're trying to make a product in a microbe, you're trying to make a product in an organism, and it doesn't grow quite as well. So you say, oh, it's toxic, making this product is toxic. Well, it's an assumption because why is it toxic? Is it stuck in the membrane? Is it not folding right? Is it producing some sort of, messing up the redox balance in some way? Regulatory networks fit in this too. We think we know. We draw diagrams that are very straight arrows that this gene interacts with this gene, this enzyme interacts with this enzyme. We think these things work, but it's actually a much more complex system than we ever intentionally did on. We draw things with nice straight arrows, and inside the cell, that's not really the way it is. And then another thing is the idea of enzyme evolution. If we have an enzyme in a pathway, it's a bottleneck, we make some assumptions that if we change the KCAT or the KM, if we evolve the enzyme, we can get out of that bottleneck. But it's not known. We just assume we can do that. On the other side, of that core knowledge, we have, things that we know, things that we can physically measure, things that we can help inform us on our decisions. And these are things like metabolic flux, redox balance, does all the everything balance out in the end, and the enzyme kinetics. So we can actually measure the KCAT and KM and say this is a bottleneck or not. So outside of the core, we have some assumptions we can make, and we have some core knowledge that we can actually measure. But now we go out, and this is really where engineering biology plays, as we kind of get out to these outer edges where we have these things that we know or we can assume. We have this core knowledge, but how are we going to improve things? How are we going to get better? How are we going to drive to new products, whether they be through fermentation or through agriculture or through pharmaceuticals? And you're left with kind of these two extremes of one, which is very search and look, and I have this down as strain search. This is where you're looking at things like evolution. Can I evolve my cell to make more of something, or to be toxicity resistant, or have some sort of improved phenotype that leads to a better product? You can use mutagenesis to do this. Random mutagenesis has been around for a long time, and you can make progress doing this old school approach. More recently, you can do things like genome scanning. And this is something that has really come to the forefront with the idea of synthesized DNA and how we approach looking for things. And this is the idea of, I could put a promoter, or I could put a terminator, or I could put a tag in front of my genes of interest, or I could look at the whole entire genome and put a promoter in front of every single gene in Saccharomyces, or put a promoter in front of every single gene in Bacillus. I don't know what I'm going to get, but I'm going to try everything and let the phenotype tell me if I made a good decision or not. So it's not. It's rational in the way that it's intentional, but it's not rational in the way in which you're getting the information back. At the other extreme is kind of where scientists want to be, where we think we're going to be when you go to think about what BioBricks and some of the synthetic biology promises to do. It's like, we're going to know everything there is to do, and we're going to make informed, rational decisions. We're going to put the pathway in. We're going to engineer that pathway through process control. We're going to engineer how the fermentation goes. So at this other end is this idea that if we know everything, then we can engineer a solution by knowing everything. They're diametrically opposed, but the way we make progress, the way that we really work in engineering biology, is if we know how to apply both approaches as rapidly as possible and take those cycles of learning, combine them, and keep trying back and forth. Keep having this Newton's cradle, go both direction, because it starts at the core and you can go either direction to make progress. So I'll give some examples here of in the past of how products have been made using almost exclusively one of these extremes. And now I'm going to go into an example of something we've done at Inscripta, where we've taken the best of both empirical engineering, irrational engineering, and combined that with hypothesis-driven engineering and made progress very quickly to make lysine. So the first example of a pure search is the example of penicillin. A lot of us are familiar with Fleming discovering penicillin, the work that they did in England during World War II to work with Pfizer and others to go from essentially a moldy cantaloupe found outside my hometown of Peoria, Illinois. They were looking all over the world trying to find an alternative to the penicillin chrysogenum they originally isolated that made penicillin. They tried to find high-producing strains. So they looked all over the world, and it was just in their backyard on a moldy cantaloupe one of their research associates brought in from the grocery store one day. And they took that moldy cantaloupe, and they not only developed a process to ferment in these large flat bottom tanks, a way to ferment and make penicillin, but they also started the first mutagenesis program, where they exposed the penicillin organism to mutagenic compounds, radiation, and they were able to then screen and find strains of the penicillin fungus that made higher and higher titer amounts. So going from essentially, milligram quantities to enough penicillin to treat every single soldier who stormed the beaches of Normandy was done purely through process control, learning how to ferment this, and through mutagenesis. There was no rational engineering in those days. Another great example is citric acid. So another filamentous fungus. And what you can see on the left here is the typical Aspergillus niger strain. It has this very typical filamentous fungal phenotype where it makes a kinetophore, it makes spores. But if you actually look at the Aspergillus niger that's grown in tanks by people like Cargill to make citric acid that is used in so many of our food products, it looks very, very different. And that's what you can see here on the right. Now, at the time, this was done through pure mutagenesis. They had two strains. One strain looked like Aspergillus niger. The other strain had this kind of binky ball, kind of clustered phenotype where it wasn't filamentous. The amazing thing to me is if you look at the genetic differences between these two strains, there are only 40 SNPs between these two drastically different phenotypes, one of which is a contaminant and one of which makes citric acid to this day. Of those 40 SNPs, one of them is a large deletion. And for years, people assume this large deletion had something to do with why it has that phenotype. And I've been involved in work that shows that it's actually not that. It's a combination of 2 or 3 SNPs that cause this drastic phenotype change to make a very valuable commodity food product. Another one, and Steve will appreciate this. This is a product that comes out of Dow called Spinosun. It's an insecticide. This one has had a little bit of rational engineering on it, but this was originally discovered back in the 80s by Eli Lilly and Company. And they had this program when everybody would go on vacation, they'd send them out with little plastic Ziploc baggies and say, hey, bring back some soil from wherever you're at, and we're going to try to culture organisms in that soil and look for antibiotics or look for interesting compounds. So when one of their employees was in the Virgin Islands at a rum factory, they just reached down, got a sample of soil from the rum factory, put it in their bag, brought it back, and they were able to culture a microbe, an actinomycete called Saccharopolyspora spinosa, and it made this polyketide spinosun. If you use, it's used as an insecticide in animal products. If you use Top Spot from Eli Lilly, it comes from Spinosun, from this product. Dow, through their joint venture to form Dow Elanco and then eventually Dow AgroSciences and now Corteva, still makes this product. They have a large fermentation facility where they're making this product. They can't make enough of it because it's still done mostly through mutagenesis and through strain selection and mutagenesis year after year after year to get improvements. And then the last one, and we're going to come back to this, is lysine. So lysine is a commodity amino acid. It's made by Cronibacterium, shown here on the left. And I have a picture of a movie called The Informant with Matt Damon here. And I'll kind of get into why this is important. But lysine and a lot of other amino acids have been for years cultivated and gone through rounds and rounds and rounds of mutagenesis and selection. And it's a highly competitive market because it's a commodity market. And you know, they've done this kind of random mutagenesis and selection. They've got strains that are pretty darn good, but in that highly competitive market, pretty darn good is not always enough. It's the difference between being the #1 dominant market player and #5 is just a couple of grams per liter. So those are all examples of products, of things that we've seen, we've used, that we're aware of in everyday life that have been all in that kind of search space. Find the natural organism that makes this, mutate it, select, screen, and find more of whatever that product is you're looking for. More recently, and like I said, with the advent of biological engineering, you start to see products that are made the other way. And probably one of the earliest examples is 1,3-propane-diol, which was a collaboration between DuPont and Tate and Lyle, where they took E. coli and they engineered heterologous genes in E. coli to make this precursor to certain plastics. It took them about 10 years and a lot of metabolic engineering, but it was purely a rational-driven example. Another one, and one that I was personally involved in, was my very first job at Amyris, where we looked at, with the Bill and Melinda Gates Foundation and Sanofi Aventis, we took a known anti-malarial drug called Artemisinin that comes from a plant, Artemisia annua. You can see it here growing in a greenhouse. And Artemisinin was a fascinating project because although it grows in greenhouse and they can cultivate it, it's a very unsteady supply. So even today, 600,000 people die from malaria. It's still endemic in so many places. And most of those are children or the elderly. They had this great drug, Artemisinin, that's derived from the plant. It was discovered back in the late 60s as part of the Vietnam War, when the Chinese were supporting the North Vietnamese. They would send their soldiers into Vietnam and they'd catch malaria. and they'd brew a tea from the leaves of Artemisia annua and give that tea to their soldiers, and that would basically kill the Plasmodium parasite. So it's been around for a while. The active ingredient has been known since 1971. The problem was the plant supply wasn't steady. You know, some years they had enough rain, they'd have a high degree of artemisinin produced from these plants. Other years, you know, because of drought or because of some extreme weather condition, they'd have less. And so the price fluctuated is anywhere from, $2 a pill to $5 a pill to $15 a pill, which, in developed nations doesn't sound like that much. But when you figure that the average person in sub-Saharan Africa spends $2.50 per year on drugs, one treatment of artemisinin is basically their entire budget. And these are people who catch malaria 2 or 3 times a year. So what we did as started out as work in Jay Kiesling's lab is he identified some of the key enzymes from the plant by doing comparative genomics. And we took those enzymes and we did synthetic DNA, we optimized it for yeast, and we re-engineered that entire pathway to produce artemisinin into yeast, in addition to upregulating the mevalonate pathway, which is already present in yeast. And through a combination of upregulation of native genes and then optimizing those plant genes, we were able to produce a strain of Saccharomyces that made 25 grams per liter of artemisinin. And Sanofi Aventis has that strain. And now what we can do is take that price that used to fluctuate, take that supply that used to fluctuate, and we can stabilize it to the point where a typical treatment of artemisinin is now less than $1. If there's a bad year on the crop, you can just turn on a fermenter and you can make semi-synthetic artemisinin to treat people so the drug is no longer short supply. One of the other important things about this too is the structure of the artemisinin molecule was such that it wasn't very stable without refrigeration. So oftentimes, and this is kind of relevant when we think about vaccines and throwing out vaccines, it's the same thing with artemisinin. If you can basically turn on a fermenter and make a supply at any given time versus having to wait for the next year's crop, that is a lot to stabilize that supply too. So we can have a constant supply chain, making up any gaps that we can't derive from the plant-derived drug in this case. But I can say this was pure engineering. There was no mutagenesis. There was no anything. Every single gene, every single manipulation to make artemisinin in this case at Amyris was done intentionally. So it was pure rational engineering. And then the last one is one that also plays, and this is kind of a popular one. And you can use either E. coli or yeast or whatever bug is your favorite, but using sweeteners, Reb M, so stevia. The idea that you can take the genes out of plants that are hard to extract the product or hard to grow enough of the product, and you can make it through fermentation. And there's been a lot of work on the Reb M sweetener to do a purely rational engineering to produce this through fermentation versus doing this through typical plant growth. So those are some examples of pure, irrational engineering versus pure rational engineering. And obviously the rational engineering has come on more strongly lately. But I guess the argument I want to make for this team and for people to think about is if you only do one, you're limiting yourself, and that you can only truly leverage products and make products rapidly if you're able to use both rational and irrational techniques. And so this is where part of my crude animation here, but this is where the Newton's cradle goes back and forth. And you can drive to hypothesis-driven engineering, and when you get stuck, you can go to the empirical engineering and back and forth. And that's where real progress is made. And that's why you're starting to see products come out of some of these synthetic biology companies that don't take 5 or 10 years to develop, but they're able to leverage both ends of Newton's cradle and make progress very, very quickly. So this is Freeman Dyson. I still love this quote. I think this quote is correct. Freeman Dyson, towards the end of his life, became a climate denier, and so I think he's not quite as popular as he once was. But this is still one of my favorite quotes, and I think it speaks to this idea of where we stand right now in this bioeconomy, and that is new directions in science are launched by new tools much more often than they are by new concepts. The effect of a concept-driven revolution is to explain old things in a new way. The effect of a tool-driven revolution is to discover new things that have yet to be explained. And so let's put that into the design, build, test, and learn cycle. So this is the traditional engineering cycle that anybody who's in engineering biology speaks to. And think about where things have gone. And I put this in the context of where they've just gone in my 20-year career. So over here in automated design, get my laser pointer here. This is a screenshot from the very first DNA program I ever used. It's called Gene Runner. It's now on the internet known as abandonware. And this is what I did my first gene designs with, was this very crude, boxy, piece by piece, maybe did some primer design editing software. And I would design genes one at a time. I would do that if I want to make a mutant, if I wanted to make a primer, it was all done one at a time by me sitting there with the mouse and clicking on things. This next example is an example of some software that we've designed at Inscripta. And it's not only Inscripta, you can do this a lot of ways, but this is the idea that design can be automated. So at Inscripta, we work with a genome editing technology called MAD7. It's a CPF1 CRISPR-based technology. And we've designed software so that if you want to go into your genome and say, design a knockout for every single gene in your entire genome, or you want to take 5 genes and do alanine scanning across every single amino acid in the 5 genes, you can use this software, and in less than 10 minutes, it can generate you every single one of those designs and say, do you want to place an order to get these designs in and do these edits? This is a software that it took me 10 minutes to learn the software, and in 20 minutes, I had redesigned and refactored the entire mevalonate pathway in yeast with a click of a button. So automated design, we understand the design rules much better than we used to, and we can design things in a massively parallel fashion. Go over here to generate. So you'll recognize the single channel pipette. This is what we all started out with in the lab. As I joined industry, as I got further along, we moved up to doing things 96 at a time. So this is a typical TCAN. And whereas the pipette was my postdoc, a lot of my early industrial work was working on a TCAN and figuring out how to scale things from 1 tube at a time to 96 at a time. And now we have instruments like the Inscripta Onyx instruments. And this, I promise this is not an advertisement for Inscripta, but I think it's a great example of an instrument that basically takes the entire generate process and automates it into a single box, where you put in a tube of cells, you put in a tube of DNA, and you come back 4 days later, and you have a library of cells that are ready to go directly into your screen or your selection. That's completely push button. This kind of instrumentation, this kind of design software pretty much invalidates my entire graduate and postdoc career single-handedly. You throw in a Q-Pix and it replaces all the toothpicks that I used picking yeast over the years. On the test side, if you guys old school recognize this is an old S35 DNA gel blot where you had 4 lanes that had to read out A, C, G, T in a pattern. Obviously, that's very different than the next generation sequencing instruments that we have now. We've come a long way from photographic plates and S35 sequencing reactions to we expect our DNA to sequencing to be returned to us overnight, if not sooner, within the hour sometimes on these next generation sequencers. Same thing when it comes to the analytical equipment, a mass spec, a GC. The very first time I ever had to go program a gas chromatographer, my samples took 30 minutes per sample. And now you look at things like rapid fire mass spec, where you're doing, thousands of samples per minute or per hour. So analytics have come a long way. And then the analysis, I come back to my gene runner software here, where I had to take my genes and go look for any SNPs or look for things that were wrong, and I had to do it by hand and by eye. Versus now we get back our information. We look at these nice little chromatographs, and we can tell right away, is this a clean construct? Does this have what I intended or not? And it's overnight versus hours and hours of painful staring at the computer screen. So these are examples of all tools that have and there's hundreds, if not thousands of more that have come on in the last 20 years that have completely changed the way we approach biology and engineering biology. They're all enabling, they all allow us to move faster and move through this design, generate, test, and learn cycle as quickly as possible. So that brings me to an example, and it comes back to Lysine in this movie with Matt Damon called The Informant. So this is basically, at the time, it was the biggest SEC bust ever, where they busted ADM, Ajinomoto, Sajaildong, and a couple other giant mega corporations who were involved in amino acid production for price fixing. So it's a fascinating book. It's based on a book. It's an okay movie, but it's a fascinating book, where basically all these companies, although they were competitors and they were trying very hard to steal each other's strains, they were also working very closely together to keep the price of lysine artificially high. And lysine is an important feed molecule for chickens and pigs, and it goes right into the importance of our food chain, and they were keeping these lysine prices artificially high. And there was a person on the inside of ADM who basically became a whistleblower for the FBI, and that's what the book and movie are about. And all the executives at ADM, Ajinomoto, were all prosecuted. And at the time, it was the biggest. It was $100 million criminal fine. It was one of the biggest criminal fines ever submitted by or ever enforced by the SEC. So there's a lot of a backstory. You know, it's pretty amazing. I'm happy to be familiar with this product from one of my former jobs, but it's important. You know, it plays into our food chain, and you know, it's if you drive through the Midwest, you'll see these giant fermenters outside of Decatur, Illinois, that are just pumping out lysine to put in animal feed. So as a proof of concept, you know, this idea that we want to take both rational and irrational approaches, you know, we want to show we can do this. How can we improve the lysine pathway? So over here on the left is an example of the genes in the lysine pathway. So there's 19 pathway genes involved in making lysine, making this amino acid. There's 20 transcription factors involved. So you can look at all these genes, and then of course there's all the other pathways, all the competing pathways, all the competing carbon flux. And so at Inscripta, using our technology, which uses a CRISPR-based technology, we said, okay, we want to go and completely look at every single gene, every single transcription factor, and then every single gene in the entire E. coli genome, and see, you know, from that irrational perspective, we want to be able to say, okay, which of these have an impact? Which of these affect lysine production? Which of these things improve lysine production? At the same time, we were rational about it. We said, okay, we know the kind of things, manipulations we want to make. We know what we want to look for here. And so we designed 25,402 genomic edits using the Inscripta editing technology. About 8,000 of those were knockouts. So, you know, the E. coli genome has, you know, approximately 4,000 genes. We doubled up. So we wanted to do a knockout in the first 25 base pairs and the first 50 base pairs. So we doubled up on them, so the odds we get knockouts. And then we looked at the iGEM promoter ladder and said, okay, let's find a bunch of iGEM promoters that have different relative strength out of the iGEM library. And let's test, you know, 5 of these in front of every single gene in the yeast gene, or in the E. coli genome in this case. And so that's what these 3,676 you know, edits are, that incorporate genes of strength from everything from, you know, 1x control to 2,547 times stronger promoters in front of every single gene in E. coli, and see how it impacted lysine. And so we did this. So over here on the left is an example of performance, you know, the fold improvement in lysine production. We were measuring lysine across the entire knockout library. And across these pathway libraries. So what you see here in purple is these are changing so took all 19 of those genes and said, okay, we're going to change every single position, every one of those 19 genes to an R, to an arginine, to you know, to a histine, to an aspartic acid. And so that's what these purple bars represent, is essentially scanning saturation mutagenesis across all 19 genes in the pathway. You know, the green had to do with some RNA manipulation. The blue are scanning stop motifs every 3rd position. So basically doing truncations across the gene. So every 3 positions, we put a scanning stop mutation and truncate the gene and see what the effect was on those. And then over here in the gray, these were the promoter mutations of the 5 varying strength promoters. So over 200,000 different types of edits were here and done essentially on an instrument. And we basically took those, we simulated them, and then we scanned them for lysine, increased lysine production on a rapid fire mass spec at 7 seconds per sample. And for an example here, this is a gene that's known to improve lysine production called DAP-A. It's one of those genes that was in the pathway. We know it's feedback inhibited by production of lysine. There was essentially almost 5,000, a little over, almost somewhere between 5,000 and 6,000 site-saturated designs that we could actually hit. We had one that was a positive control. We know if we hit this 84, this glutamate here at 84, and changed it to a threonine, it had an impact on lysine production. And then we also had the promoter insertion. So we basically had a control built into this, So we looked at the control, made sure it showed up, but then looked broadly. And so what you can see here is we start to look at some of our outcomes. And so over here in the blue, so we had this positive control. We knew DAP-A with this particular mutation would give us an improvement. So this is where our positive control is. Over here in the blue is where our wild type was. And so when we look at those genome-wide knockouts, we see some hits in the genome-wide knockouts in the wild type background. They get a little higher when you go into this DAP E84T background. And we look at the promoter swaps. And we say, okay, look at the promoters. Same thing. We have some hits in the promoter background, in the wild type background, that look like they probably also translate over here in this positive control background. But now we say, okay, we're going to start looking wider. We're going to start stacking things. So for example, what if we take that promoter hit and combine it with that, E84T hit? And we go from essentially over wild type 460x higher production. Now we're almost to 1000x. But that's still not enough, because now we start looking at these other things we went after. So we add that promoter hit with a different promoter hit, and now we're seeing 10,000-fold improvement. And now we say, okay, we're going to look at another gene hit we had, this in Glen D, and another promoter hit. And all of a sudden, in basically 5 weeks' time, we go from minuscule amounts of lysine to lysine being produced at 14,000 times higher production, fold improvement over the wild type gene. And this is through this both, taking this approach of, hey, let's take some things we know, let's take that rational approach, but then let's look everywhere in an irrational approach, combine those things and see how fast we can make progress. And so in 5 weeks, using this technology, using this combination of rational and irrational approaches, we were able to make a lysine strain that is 14,000x improved over where we started. And so you put all those together. You think about the proteins, you can look at the protein level and do scanning mutagenesis. You look at the entire pathway and how you manipulate the entire pathway. And you look at the entire genome. And taking all three of these approaches, what you can do is we generated 200,000 edits. And what we have here is all these positive edits. And we have them in two different conditions compared pairwise. So these are all the genes that if you hit them, they make a difference in lysine production. And in this case, we basically have the fold improvement if you're trying to do it in two different mediums. So minimal media, which is kind of what we grow the E. coli in, versus lysine production media. You compare those two. And in some cases, you'll get hits that only work in one media. Other cases, you'll get hits that work in the lysine production media. And in these cases, in the upper right-hand corner, we see hits, we see improvements, we see combined edits and stacking that show us this improvement that's 1,000-fold in one case, 100-fold in another case, that really change the production value for making lysine. So with that, I kind of want to pause. I'm happy to stop there. I want to make sure I have time for questions. If there's any questions at this point, if not, I have a little extra. But I want to make sure we have some discussion too. So,
Patrick, thank you so much for the presentation. And again, I think the cautionary tale of not being able to trigger back and forth between rational and irrational approaches to be able to drive and accelerate. Also, I think for me personally, the evolution of the underlying technology that enables this and the rate of change that's taken place, as you mentioned during your talk, it was also very enlightening. Again, I think that Patrick's already had me ask the question, why didn't we put that on our equipment list? And they said, well, it didn't exist 2 years ago. So again, I think the opportunity to move forward. I want to encourage questions from the participants. Please use the Q&A function at the bottom of the screen. It's between the participants in the chat to post questions for Patrick, and we will. I will take them from there. I guess one of the first questions comes from somebody that's worried, or not worried, I shouldn't say. One of the things that we've talked about for a long time was the idea, this was originally called synthetic biology. Now it's called engineering biology. And what about consumer acceptance, public acceptance of the idea of rational and irrational engineering. Thoughts, Patrick, on the perception of this technology as it relates to its application and acceptance by the market?
Yeah, so I mean, clearly we've been using irrational engineering for a long time. People are comfortable with that. People take penicillin or its derivatives when they need to. People eat lysine when they eat bacon, whether they know it or not. So I think on that side of things, that's considered, or people are comfortable, and that's considered a natural process. It's on that example of that rational engineering where you're doing intentional manipulations that people may get uncomfortable. Although if you make products like, hey, you can die from malaria or take this drug that was made through engineering, people are probably going to take the drug. But I think you have the split. I mean, we look at people who are against vaccination right now on the mRNA technology. There's always this distrust of technology, and the best we can do is show that it works, show that it's safe, and it's a process of education. Our ability to make things faster, to make improvements, whether it be to address climate change, whether it be to make products that are coming from petroleum and make them through biology. The more we show the value in what we do, the more we show the value in moving rapidly and what biology engineering has to offer, the more examples of that we can get, the more comfortable I think society will get with using those solutions.
Yeah.
There's a great product made by Amyris called Squalene. And since they started making this product from a C30 molecule, they started making it through making farnesene and putting it together in yeast. The amount of shark fishing where squalene was gotten, where normally squalene comes from shark liver, the number of sharks that have been saved are in the millions and millions of sharks because we're making a product through biological engineering that before was sourced through a very destructive practice of harvesting sharks and killing sharks. So there's those stories that we got to tell.
Yeah, Patrick, and I think that is actually a great example because wasn't Pat Cavello from NRC PBI involved with that? Would be the link back to Saskatoon?
He was involved in that. He was also the person who in Saskatoon discovered 2 enzymes in the artemisinin pathway that we weren't using. And then when he licensed that to Amyris, we put those genes in the pathway. We went from basically making 2 grams of artemisinin to 30 grams of artemisinin by using the research he had done up there in Saskatoon.
So that's a good plug for why the cluster in Saskatoon is the center of biological universe. So that was unscripted, folks, just for a reference. And I think I think it's a great question about the cautionary tale of having to be thinking about proactively, not only the technology and the innovations that the technology can drive, and there's some other questions that we'll get to in the Q&A in a minute, but also the need to be thinking about the regulatory framework as well as end user and consumer acceptance and being proactive in trying to position and engage so that you're technologies that can have a significant improvement on quality of life, environmental sustainability. Your examples of artemisinin and your examples of squalene are, that those opportunities are not squandered and not lost on being able to contribute to improved sustainability, economic, environmental, and social. And again, I think that's a really good point. Another question relates to your example around lysine. Why did you peak out at the 1,400-fold improvement in productivity? And again, the timeline would also be another question, like how long did that whole process take to go from not just the 5 weeks in round 2 and round 3, but also thinking about it from the initial start to those 14,000-fold improvement?
Yeah, so So the Inscripta technology, which is based on CRISPR technology that incorporates not only CRISPR but also a barcode, so you can basically screen massively in parallel in a pooled format. You're not talking 96 well if you don't want to. You can do a selection and things like this and still identify what your hit is. So typically, these libraries, this design work, that software is made it such that those 200,000 designs in the software maybe took a couple hours. of crunching. So there was very little human input. It was just based on algorithms to design it. It took a couple of weeks to generate the oligos, to generate the material. The instrument, generates 10,000 hits at once, and it takes 48 hours for E. coli. So to actually do all the instrument runs and run all 200,000 was a couple of weeks of that. So it was maybe 2 or 3 months of buildup time and then execution. And then after that, that 5 weeks of showing, okay, here's the screen, here's the stack, here's the screen, here's the stack. And it's this idea that you look very, very broadly, be very expansive in your search, and let the phenotype come back to you, let the science come back to you, and drive where you want to go. There's an example on lysine where the ADM strain they've had it since like 1985, and from 1985 to 2015, they just took it through rounds of mutagenesis, and over years they basically generated 2,500 SNPs. But when you go back and deconvolute that and see those that are actually contributing, there's only 20 of those that actually had any impact whatever. So most of when those irrational approaches, you're getting a lot of garbage in there too. This approach allows you to look at all 200,000 or 2,000 SNPs or 10,000 SNPs or whatever like that, but then very quickly deconvolute and say, okay, these are the ones that are important, these are the ones that are not. I think the other thing that's kind of missed from this technology as we swing back and forth, and I think it's something that scientists need to be aware of, is we think very linearly. And this is something that I think we can get ourselves out of using these new tools. And this is the idea that you go and you test a hypothesis. And if it's positive, you carry it forward. But if it's negative, you tend to drop it. You say, okay, I tested the idea, it didn't work. However, context changes. And so as you get one positive hit, your ability to go back and look at old ideas in a new context, we very rarely do that. It's hard, it's difficult, it's like you've already dismissed that idea. We want to think very linearly and very forward ahead. The tools and technology we have now say that's not hard anymore. You can go, you can test something. If it works, great, you carry it forward. If it doesn't work, a little bit later on, you can really easily reapply that same library, that same idea in a new context, and find that all of a sudden now it makes a difference. Now, because you didn't have that change the first time, you couldn't see it, but now in a later context you can. And I think that's one of the things that really drives forward this technology too, is making sure we don't exclude ideas just because they didn't work early on.
I think that is an incredibly important comment for not just doing the research, but when you think about the innovation pipeline, there's lots of inventions that are very good that require, but they'll never be commercial products because they require other innovations to make them. So the context has changed. So I think that's a continuous learning cycle that I think we need to embrace, and the ability to do work at the scale you've described in your lysine example and other examples that you've hit on during the course of your talk, I think is a real enabler for us to, not shut down doors and venues, and again, being able to go back. I think that ability to reuse ideas is good. Another question about your experience: is it at the structural level of the pathway, the enzymes in the pathway, or more on the regulatory? Do you see a difference, or is it just in terms of where the biggest bang for improvement is, or is it more, are you going to answer it like a lawyer and say it depends?
They're all important. It goes back to, what I was saying, is you have to do both. So the regulatory aspect of a gene, or turning things on or turning things off, or turning things up or turning things down, is important. And to be able to control that, I think is a tool that we can use. Changing the enzyme kinetics is also a tool that we can use. If you're only using, what's the saying? If all you have is a hammer, everything looks like a nail. Right. So that's the case there. I would say that yes, we want enzymes that work more efficiently, and I've worked on enzyme. I've done enzyme evolution. We want them to work more efficiently, and that's the case. But other cases, you still need to have the enzyme around. Regulatory becomes important for a vital enzyme you can't get rid of, but it has some feedback inhibition. Well, maybe you can turn it down and get kind of the minimal effect. And so both are, there's a push and a pull. And if you're only doing one, you're missing opportunities.
Okay, great. Thanks. I got a question from somebody why has there been a name change between synthetic biology to engineering biology? And I'll put a plug in for our next speaker, Steve Evans. He can provide some personal experience on that. But Patrick, why did the naming evolve?
Yeah, it's basically social media type thing. When you think synthetic, you think synthetic chemical, synthetic rubber. It has a certain sound and a certain connotation to it, where, oh, this is not natural. So even though you're using natural genes and you're using biology, you're applying biology is not natural. If something's not natural, there is a visceral effect with it in some people. Versus engineering, you're like, oh, that's making bridges. That's very rational. It's very process driven. Very mathematical. And so I think, biological engineering means that you understand what's going on versus synthetic biology if you're doing something unnatural. That would be my take on it.
So another question is, that's great. And I kind of think, again, it's the connotation. And again, lots of times when naming is left to really smart scientists, they are not the best marketers in the world. And I think that's especially in the ag and food sector, we need to be very aware very early on in our innovation cycle of the impact of the regulatory framework as well as end user and consumer acceptance, because it's a major barrier. And Patrick mentioned some of the challenges we have with the mRNA vaccines for COVID, which again represent how thinking can get in the way of solutions that are valuable to everyone.
Yeah.
Couple questions.
Actually, just real quick, I know we're close to the end of the time, but I do. So I want to answer the questions, and I'm happy to stay a little bit afterwards if we're able or whatever like that. Yeah. But I do have a couple more slides I really want to share because I think it's important.
Yeah, let's go to those because I've seen them and I like them. And one thing, while you're pulling your deck up again, folks, what we are going to do is with the questions you've typed in, we're going to provide written answers back. We'll put down what Patrick has answered, and then we'll run them by Patrick so we can give everybody that's been on the call a copy of the questions that were asked and our answers, just so that we can kind of see where we are as we go through this webinar series. Wait, there's more. Patrick, please.
So I wanted to bring it back to this idea that, Steve and I have both worked in plants. My background's in botany. I've shown a lot of microbe stuff, and that's, people often get like, great, microbes are nice, fermentation, but I work in plants. How can this approach, not the technology, I'll give an example of the technology, but what are the tools, what are the approaches that I can use in engineering biology in the plant world? So what we have here is an example of Bacillus thuringiensis crystal proteins. And you can see from the corn example here, the top one is a GMO that has a cry protein built into it versus the bottom one doesn't, and it's susceptible to larvae in this case. So when I worked at Dow, we were looking for a lot of cry proteins, and they had a process where they had a bunch of Bacillus thuringiensis genomic DNA pooled in 96 well plates. And this was their process. This is how they found cry proteins, is they did not use automation. They used multi-channel pipettes, and they would do degenerate PCR, and they would do about 5,000 clones of this a year. They had a team of 3 people working on this, and they do about 5,000 clones a year. And they do this degenerate PCR, and they look for bands and say, okay, well, this band is about the right size of a cry protein. We're going to clone it into bacteria. From these 5,000 candidates, we'll get, a couple hundred. We'll feed those to insects in our insectarium. We'll express it and feed it to insects. And then with this process, they would do, 1 to 2 new cry proteins every year, and maybe 1 or 2 of those would then actually make it into a product. It was very labor intensive. It was very time consuming. It was very people driven. And I even remember at Dow, I was like, well, instead of doing this by multi-channel pipette, can we do it on a robot? And the person in charge says, absolutely not. Don't even mention that. I'll lose my resources. I can't imagine doing it a different way. This is how we do it. We don't want to change. So that really had a big impact on me. And it's like, well, we have to change. If we're gonna proceed and move forward, we have to do it differently. And so I'm not sure this is right, but conceptually, I just want to put this out there for discussion of, hey, what if you use this new technology, these new tools we have? What if we use genome editing and do wide searches? And so let's have a library of CRY proteins, of CRY winners, of novel CRY proteins. Don't worry about the generate PCR, just pull them out. CRY clones that just missed. There was a ton of things that had early wins, but they didn't quite make it. And so let's take those, and instead of doing it one at a time, let's use something like the Inscripta technology, where you basically take that entire pool of CRY proteins and you do massively parallel mutagenesis on it, where you take each individual winner, you do saturation mutagenesis, you run it through something like the Onyx instrument, and now you have these pooled cells, and you feed these pooled cells to an insect and look and say, okay, which of these pools actually have efficacy against the insect? And now you can use the barcode and very rapidly pull out through a gut microbiome look. Those barcodes will say, oh, hey, this particular design is really enriched in these cells that died. So it's a high throughput pooled methodology that you can leverage in this case. And again, the technology is not worked out. I'm just trying to prompt this idea of think differently. And all of a sudden now you can run this QC assessment. You look at your barcodes and say, okay, here's a design that looks really interesting. So you're using new things, you're using old things, but at the end of the day, you're getting a corn product much faster. You're pushing more things through your pipeline. You're screening more, you're testing more, and you're getting potentially better products. You're not throwing out products that were close, but you can actually take those and make something out of it. So I just wanted to share that. I don't know if this is right or this is wrong, but tools aren't only in bacteria. I talked a lot about bacteria. I talked a lot about yeast, but there are new ways we should be looking at every technology, whether it be agriculture, whether it be pharmaceutical, or whether it be biological engineering. So I just wanted to put that out there as kind of food for thought.
Thanks, because again, I think it's the challenge is it's good to embrace change as opposed to being comfortable where you are. Ask the question. And again, change, some organizations fear it. We need to embrace it because the challenges that we are facing, particularly around how do we produce food in a sustainable manner to feed a growing world, it's been exacerbated by COVID-19. It's showing that we've got a lot of work to do, and we need new ways, new tools, new approaches. One question: when you interviewed at Dow, you described engineering biology as A, B, and C. And it wasn't the Jackson 5 you were doing. So what does A, B, and C stand for? And I want to delve into C a little bit because of one of the questions that came up.
So I'm trying to, it's been a few years,
Well, I know it's been a while. C.
So I often refer to A, B, and C as, it's, what is it? It's
automation.
All right, give me a second here. Let me gather my thoughts. So the idea is, it's ABC. It's 1, 2, 3. It's not 1, 3, 2. You can't skip to the end. You can't, go immediately out of order. So you have to have. So there's a lot to do with process. It has a lot to do with order of operations and how quickly you can cycle through those order of operations. So the idea that you can in the terms of people, I always think that when people in organizations are going to through organizations, you have the people in the R&D at the front end, and you have the people on the back end who are doing process, who are trying to scale, who are trying to produce or grow or whatever like that. And the part that you usually skip is number 2, which is going from that R&D effort into an actual scalable process. And I think that's one of the hardest things to factor into this. Now, what's interesting about that is a lot of that learning aspect, a lot of that in-between aspect can be facilitated through data management and through looking at large data sets. So early in my career, it's like, okay, I have all these rational ideas. I give it to the process engineer. They hopefully made it work, but we didn't do a lot of process with that. What we find now is with these larger data sets, that in-between part, that data management, that information management part becomes more and more important and factors into decisions we make. So we don't have to go straight from 1 to 3. We can go from 1 to 2, process that large data set, and it can often inform us on what the next steps are. So in the case of, the lysine example I gave, we went through that first process, screened a lot of things singularly, and then we started stacking them. But we also know from that data set, like because of epistasis, if you stack things that are exactly the same, so if you keep stacking on DAP A, you can only make so much progress. But through looking at the entire data set and saying, okay, what are the things that are most distant? What are the things that are less likely to be epistatic and more likely to stack? And I think that is another part of it that is more and more prevalent in the tool area, is that data management, that data prediction.
Perfect. Anyway, Patrick, I want to thank you on behalf of not only the Global Institute for Food Security. I want to thank you personally for being the first out of the gate. This was a fantastic talk. And also for our other partners and sponsors, the work that the team at Ontario Genomics is doing to spearhead our national engineering biology strategy here in Canada. And again, thank you so much, Patrick. And we will capture all the questions and share with Patrick in the Q&A, and then be able to provide written responses to the participants. Thanks for everybody for hanging in there. Thank you for your questions. We will get answers to them because there's a lot more than we were able to cover. And I think I might have had more fun than anybody else today, because that was a fantastic presentation, Patrick. Great job.
All right. Thanks, everyone.
Okay, take care.